All right. Hello, everyone. I'm Debbie. I'm from the Center for Aquaculture Technologies, and it's a pleasure to be here as part of See the Future 2026. Today, I'm going to share with you about how genetic innovation, and particular genome editing, can help strengthen and productivity and resilience in aquaculture. So, as I'm sure many of you are aware, aquaculture is already a very fast growing source of animal protein in the world, and it will be very, very important to feeding the growing population in the years to come. But that future growth can't rely on scale alone. It's going to come from smarter, more resilient, and more sustainable production systems. So at the Center for Aquaculture Technologies, we commercialize genetic technologies to improve the productivity and sustainability of the aquaculture sector. Our work spans several disciplines, including breeding, genotyping, genetic data analysis, and of course, genome editing. Our focus is on responsible commercial scale deployment of genetic technologies in partnership with producers. Aquaculture producers everywhere are facing increasing pressure coming from climate change and climate stress, disease challenges, environmental challenges, are increasing biological risk. At the same time, environmental expectations demand a lower footprint and stronger containment. And of course, the global demand for protein and specifically for seafood continues to rise, especially in food insecure regions. So the challenge is not simply to grow, but how we grow and increase productivity and resilience and sustainability at the same time. Genetics is a lever that we can employ across agriculture. The most consistent driver of long-term productivity gains has been the development of improved genetics. So we're talking about plants or animals, and aquaculture is no different. Conventional selective breeding has already delivered major improvements in the traits that we need in food production, including growth, yield, and overall robustness. Genome editing represents the next generation of breeding technology. And the nice thing about it is it integrates very well into existing breeding programs as a tool. It's not there to replace them. What it does is it allows us to accelerate the genetic gains and the traits that we're interested in that breeders are already pursuing. So what is genome editing? Really briefly, genome editing recreates genetic changes that um, can occur naturally through natural breeding processes, except it does so in a more efficient and precise manner. It's important to note the key features in this case are no new DNA is added. So in the conventional sense of the term GMO, it is not considered to be a GMO. What it does is results in a faster and more predictable path to achieving the beneficial traits that we're looking for. Skipped one. Genome editing can be applied across traits. It's a tool that can be used to deliver a variety of solutions that matter to all stakeholders, including the producers, the regulators, the consumers. These include characteristics that support productivity, animal welfare, and sustainability, like growth, yield, feed efficiency, and disease resistance. And we can even use it to introduce traits that are not possible to select for using conventional breeding methods, like sterility. The key advantage of genome editing is speed. It dramatically accelerates genetic improvement relative to conventional selective breeding methods, which build gain each generation. Genome editing can introduce a significant improvement in a characteristic in a single generation. And the nice thing, again, is that it works within established breeding frameworks and can create significant value in a short amount of time. We're already seeing this impact in commercial aquaculture species. In tilapia, CAT has created single gene edits that have delivered substantial improvements in growth performance. So in this example, we tracked growth performance of edited and tilapia for growth relative to their non-edited siblings. And in our hands, we're able to demonstrate up to 48% improvement in growth performance, which is significant and means it's representative of a meaningful shift in farm productivity. In another example, we've created single gene edits that can have improved yield. And in the full sibling comparisons you see here in the pictures on the left and the right, we've highlighted the edited and the non-edited animals for you. And you can see the edited animals, significant increase in the edible portion, the muscle component of those tilapia. And so that translates directly into improved outcomes at harvest. But the productivity traits alone were not enough for us, and we've spent quite a lot of time thinking about responsible deployment of significantly improved productivity traits. And for us, the sustainability and resilience of that system is just as important as the productivity. So what genome editing has allowed us to do is reliably produce 100% sterile fish for aquaculture. It's a critical enabling technology when we're introducing these significantly improved traits. They're very beneficial in a farming environment, but they may not be as beneficial in wild populations. So it's really important for us to make sure that those animals with those traits are reproductively sterile. Sterility addresses multiple challenges at one time. So you do get improved growth benefit in sterile fish because they're reallocating their reproductive energy to growth. It encourages investment in technologies like these by protecting proprietary genetics, prevents escape farm fish from reproducing with wild fish, and addresses environmental and regulatory concerns. And so for us, sterility is foundational for delivery of sustainable aquaculture innovation. The really exciting part is that genome editing is no longer theoretical or simply a research tool. Genome editing has now been applied at commercial scale in tilapia. That work is being done by ourselves through strategic partnership with a company called Brazilian Fish, located in Brazil, and where we're working to deliver genome editing at commercial scale. A key feature that's enabled us to do this is the development of our mobile laboratory. So the mobile genome editing laboratory allows us to do precision genome editing farm side, as long as we've got some power and a work surface to work on. We are working directly to integrate edits into our partners' breeding program at scale that enables them to maintain their existing, continue to improve on their existing genetic diversity. And the mobile lab is really the key feature that's enabling us to deliver genome editing at commercial scale. So we announced the partnership with Brazilian Fish just about a year ago now. So what have we been doing for the past year? So in 2025, we were able to make several trips with the mobile laboratory to Brazil. We've injected more than 25,000 embryos with enzymes or genome editing enzymes for sterility, growth, and improved yield. Thousands of edited fish are in tanks in Brazil now and growing up as broodstock. The regulatory determination process in Brazil is underway, and we're optimistic that commercialization of the progeny of these broodstock can be expected later this year. When we apply genome editing at scale, the value is significant. And we've done some modeling of the value that genome editing can bring to several different species markets, either through decreased cost of production or increased revenue. Here I've got an example where in Brazil, we can show that genome editing has the potential to deliver hundreds of millions of dollars per year to stakeholders in the tilapia market. That value comes from improved growth, performance, feed efficiency, and fillet yield, and represents a transformational shift in aquaculture economics in the tilapia market in Brazil. When we scale it up to the global level, the implications are very profound as we consider, feeding the future. Across species, editing even a single trait like yield can significantly increase the amount of food that we're producing without having to expand our environmental footprint at all. So that is a direct, we can draw a direct correlation to global food security and sustainable industry growth using technologies like genome editing. So summarizing everything, in our view, genome editing will not replace breeding programs. It becomes a tool in the breeder's toolbox. The sterility is key to unlocking the responsible deployment of genetic technologies like these that do make such a profound impact in the characteristics of the animals. Over the next 5 years, we feel that genome editing has the potential to dramatically increase productivity with deployment of traits and solutions like the ones that Kat has developed, and ideally reduces the required resources to produce more seafood that we all would like to have. So with that, I will say thank you so much to all of you. Thank you to our collaborators and partners who are helping us to bring this gene editing innovation to commercial reality. If you'd like to contact us, you can visit us on our website. My contact information is on the title slide. You can reach out to me directly with any questions or discussions that you may wish to have. Thank you so much, and I hope you enjoy the rest of the meeting.